Well, thank you very much for the introduction. Thank you to the organizing committee for the opportunity to come and speak here. This is my second time in Montpellier, so it's wonderful to be back, this time in the winter. So a little different than the summertime last time I was here. And so, as Frank said, I'm an engineer by training. I come from chemical engineering. All my degrees and all through my postdoc was in chemical engineering. And I started working in this field as a postdoc, working on microfluidic models, and I'll talk a little bit about some of those here as well. But I've kind of moved over the course of my career towards more and more clinical application. And I think what Frank said is right on. It's very useful to be able to talk across disciplines. And so I have several collaborators here. Aaron Fogelson and I have worked together for, I think, over 15 years, who's a mathematician. Pierre Majan, who I've served on committees with, is a biologist. And now I serve in a hemophilia and thrombosis center that treats genetic bleeding and thrombotic disorders. And so it's a lot of work with clinical samples. So I've been able to do quite a bit coming from kind of humble chemical engineering background. So what I want to cover today, and hopefully you'll be able to walk away with some of this information, is first to just define hemostasis and what are the sub-processes involved in hemostasis. I'll do this kind of from the lenses of the biophysical phenomenon that regulate those. And so I'll talk about the forces and flows that regulate hemostasis across different length scales. At the end, I'll talk a bit about somewhat maybe a little bit of an unappreciated part of this is how blood flow regulates coagulation reactions and kind of the mass transfer regulation of coagulation. Throughout the talk, I'll talk about some different experimental models that can test and measure some of these biophysical phenomenon. And at the end, I'll talk a little bit about how those could be applied clinically. And finally, some emerging approaches and concepts that hopefully you all will work on or be at least inspired by. So I'll start with this figure that Aaron and I worked on a review article in the annual reviews of fluid mechanics in 2015. I started a lot of talks with this because I think it's a nice introduction to these two sub-processes of hemostasis. And so the first is going to be platelet function, the second will be coagulation. But for the purposes of platelet function, we have all these phenomena and adhesion to the vessel wall or to the subendothelium of the vessel wall, aggregation between two platelets, activation, and then finally contraction of a fibrin clot. All those things, in addition to VWF on Willebrand factor, which is a large multimeric protein in the blood, they're all regulated, they're force-dependent phenomenon. And so understanding and quantifying the forces is really significant for all of those processes. So I'll talk about a big chunk of the lecture. We'll talk about those different forces and flows. Once a platelet does become activated, and that can happen through a number of processes, a number of, let me get this, there we go. Through a number of interactions, either through signaling, through these receptors that are either interacting with the endothelial matrix underneath in the vessel wall, but also some soluble agonists, things that are secreted from platelets like ADP from granules or thromboxane that's secreted and generated inside of the platelet, or thrombin, which is the end product of the coagulation cascade. Those create a number of activation-like substances. So when I teach this to my own lab or when I teach it to medical students, I always talk about platelet function as being a spectrum of activation. So there's a number of events that happen, sort of the first being activation of integrins. So this one in particular, alpha-2b beta-3, that's the highest expressed protein on platelet surface. So there's usually about 100,000 of those on each platelet. That goes from a low affinity to a high affinity state where it can interact with fibrinogen and fibrin. The next sort of stage of platelet activation would be granule secretion. Hundreds, maybe thousands of things are secreted from alpha granules and dense granules. One of them is ADP. So there's this autocrine signaling through these P2Y1 and P2Y12 receptors. And then finally, at the kind of latest stage of platelet activation is the exposure of a lipid called phosphatidylserine, that's when it's almost like an apoptotic-like mechanism where platelets express this negatively charged surface. And on that negatively charged surface, you could have these coagulation reactions. And so this is a little bit of a different model if you're used to seeing what is often called the cascade or waterfall model of coagulation. That's where you have an intrinsic pathway and an extrinsic pathway that lead down to thrombin. This is a different model or a different concept of coagulation, which is called the cell-based model. This was introduced by colleague Mac Monroe and Maureen Hoffman kind of at the end of the 90s, 1990, 1999, 2000, and then converted into a mathematical model by Dr. Fogelson here. And this is, I prefer to show this when we're talking about hemostasis or what's happening in vivo, because this is how these reactions happen, not in a tube ex vivo, which is how we test the PT and APT time, APTT time in a clinical assay. But this gives you a sense of where these reactions happen. And what's important to note is that they generally happen on surfaces. So some of them happen on the surface of endothelial cells. Many of them happen on the surface of activated platelets. And so platelets are really serving as this catalytic site for coagulation reactions. And I'll come back to some of the details of this reaction, but suffice to say this is a very highly nonlinear network, and it's one that is very hard to intuit without the help of mathematical and computational models, given all the feedback and feed-forward mechanisms. And so these are rate-dependent phenomenon, and so I'll talk a little bit about how blood flow and mass transfer in particular can regulate coagulation following primary hemostasis with formation of a platelet plug. So for the big chunk of the talk, I'm going to kind of walk through hemostasis from the perspective of different length scales. And so we have a number of phenomena that are happening from the vessel scale to the cellular scale to the molecular scale, which, there we go. I'll start kind of here with platelet margination. I'm having a hard time with this. And move my way over until looking at adhesion and aggregation of platelets, so things that start on the centimeter length scale, go down to the cell scale, so sort of the micron scale, and then finally a little bit about some of the mechanotransduction or mechanosensing mechanisms that platelets have that happen on the sort of nanometer scale or the molecular scale, if you will. So I think based on the posters that I saw out in the atrium here, many of you are familiar with the rheology of whole blood, but I do like to always start with this, and this is a kind of classic experiment. This was done by Xu Chen. It was published in Science in 1970, and it's still to this day probably one of the more elegant experiments ever done in rheology, where he was looking at the behavior that had been observed is that we have, this would be whole blood. Y-axis is the viscosity relative to water. You can see the shear rate on a log scale on the x-axis. And there's clearly a shear thinning type of behavior, but it's a little bit different because there's also a known yield stress with whole blood. And so this is an experiment where the platelets were either fixed, and so if they're fixed with glutaraldehyde, that's the green line here, and that gives you the idea of deformation. So deformation of red blood cells is what gives the whole blood the shear thinning behavior. But also there's this aggregation or the formation of rouleaux. So they look like this, these kind of like stacked red blood cells. And that's mediated through large proteins in the plasma, mostly fibrinogen. So if you resuspend red blood cells inside of a buffer with just albumin, so you don't have all the other plasma proteins, you also get this reduction, sort of a order of magnitude reduction in the viscosity, and that's because they don't form these rouleaux. So the rheology of whole blood is unique in the sense that you have both this yield stress that you have to overcome. So as you go, to not very high shear rates, but you start to break these apart. They're just held together by colloidal forces. And then as you get to these larger shear rates that you would expect in, say, arteries, you see this flattening of this. But I think it's somewhat important in a lot of the modeling that folks do, because generally often people treat blood as a Newtonian fluid. And it does act Newtonian in one part of that curve. However, you can see that it does have a pretty significant change. And when you have parts of your flow that have low shears, whether that's if you're modeling something like venous thrombosis, the shear rates inside of the venous valves are quite low, or an aneurysm, or anything where there's any sort of pooling or cessation of blood flow, you'll get the formation of rouleaux and have a pretty significant difference in the viscosity. So that's one, I think, important concept to always remember when we're talking about blood rheology, is that it's non-Newtonian and it has these two phenomenon. Another is that the rheology of blood depends on the size of the tube that it's going through. And so this is known as the Farias-Lindquist effect. It was first described in 1919. And this is a phenomenon by which as you move from a large diameter tube or a vessel down to something that starts to approach the diameter of a red blood cell, which for humans is 8.5 microns, you see this significant decrease in viscosity. Essentially, this is a steric argument because blood cells can only get so close to the vessel wall. They're going to move away from it. I'll talk a little bit more about that and the implications on platelet margination. But there is this decrease in viscosity as you move down from something that's on the order of, let's say, 1 millimeter down to 10 microns. And then as you get into the capillaries, you have this squeeze-like flow. There's a significant amount of friction between the red blood cells and the glycocalyx, which is this almost gel-like thin layer on top of endothelial cells, and then you see this very sharp uptick in the viscosity again. So those things are very important. Understanding that it's a non-Newtonian fluid, and that the rheology is highly dependent on the size of the channel that you put it through. It's also important for a lot of experimental models that are used now, especially microfluidic models, because what people who work in this field might appreciate is that most of the microfluidic channels are sort of on the steep part of this slope, which means that when one person does an experiment in a 50 micron channel and another person does an experiment in a 150 micron channel, they could be experiencing very different viscosities, and that can really make the interpretation of those experiments a bit problematic. These are just this bullet-like shape that you see is characteristic of going through capillaries. Okay. So now I'll talk about platelet margination. Platelet margination is the phenomenon by which platelets get pushed towards the vessel wall. Obviously, that's important because you want platelets near the vessel wall where they can do their job in hemostasis. And so the kind of physical phenomenon that creates this is that we have this anti-margination, or this migration of red blood cells away from the wall. They take on this type of structure, this kind of like slipper-like shape that gives them almost like a little bit like an airplane wing. They have a pressure that pushes them up off the wall. And so as they anti-marginate and push towards the center of the vessel, they're going to create this gradient in red cells. And so there's more red cells in the center than there are towards the edge. So that creates a gradient, and that gradient, in addition to sort of pushing things out sterically, there's going to be this platelet collisions between red cells. That's going to bias their motion away from the core, where there's going to be a lot of collisions, towards the vessel wall, where there's less collisions. And so that's just kind of an enhanced diffusivity, and that's the way that it's modeled. But it turns out that if you just have a single diffusivity, you can't really predict the kinetics very well of platelet margination, so how quickly a platelet would go from, say, the center of a vessel towards the vessel wall. And so most models involve some other type of drift force. One that's quite common is the idea that there's this tank treading motion. This is required for red blood cells to stay in this conformation and not tumble. And so this creates a little bit of a microflow that will almost act like a little conveyor belt, such that platelets get pushed into this cell-free layer or red cell-free layer and stay there. And so at the end of the day, what you end up seeing if you looked at the distribution of red blood cells and the distribution of platelets is they tend to be sort of the inverse of each other. So this high concentration of red cells. And the flat profile's important here because that's that yield stress that I talked about before. So at the very center of the vessel, of course, the shear stress is 0 or very small as you move away from the center line. And so you have essentially a solid, if you will, until you get above the yield stress. And the enrichment of platelets, it's about the 3 to 4 times higher near the vessel wall than it is in the core. And of course, that has physiological significance because you want to have rapid platelet adhesion if there is a vessel injury at any point in time. Okay, so now I want to do a case study. This is work that I did with Alyssa Wohlberg and Aaron Fogelson several years ago. And this was to look at the effect of hematocrit on platelet accumulation in a couple different experimental models. And so one of them is to have a model of high hematocrit in a mouse. And so what is done is we take blood from a mouse, separate out the platelets and the leukocytes, wash the red blood cells and resuspend them at a hematocrit of 70%. And then we inject those into another mouse, and then wait 24 hours, essentially elevating their hematocrit, or elevating the fraction of cells that are red cells, and then doing some measurements. And so this is, what we get at the end of the day is we get the normal hematocrit in these mice is 0.39. And so by doing this injection of exogenous red blood cells, we can elevate it to 0.47. So it's a modest increase, but we still see some significant differences. And so what you're seeing here is the results of a very common arterial thrombosis model, where you take some filter paper with ferric chloride on it, you place it in contact with, in this case, the carotid artery, hold it on there for a certain amount of time, and then with a Doppler probe, measure how quickly the occlusion forms, or how quickly the blood flow stops. And so you can see that these are the control mice, and that when you increase the hematocrit, you decrease the time to occlusion. So essentially the clot is forming a bit faster. In this case, you can see that the onset time about the same, and then the kind of formation rate, so how fast the Doppler curve changes with time, is a little bit faster in the case of the hematocrit. So again, this is a pretty modest difference. It's just the difference of 0.8 or 0.08 of the hematocrit, but you still see this significant difference in the occlusion time in this particular animal model. We also have did the human version of this in a microfluidic device, and so we take human blood, we split it into essentially platelets and red blood cells, and then resuspend the platelets in the plasma at a slightly higher hematocrit. And so it turned out to be about 41% in the human, and 41% was kind of the normal. We were able to get it up to about 54%. And so this is accumulation of whole blood on type 1 collagen at a shear rate of 750 reciprocal seconds. And you can see that this spot's a little bit brighter. Oops, go back again. Yeah, this spot's a little bit brighter than this spot. The quantification is here. So you can see these are the average of the case where we have the high hematocrit in the black dots, and the normal hematocrit in the non-filled in dots. And we can look at the kind of lag time, how long it takes for the clot to onset, but also the rate at which platelets accumulate. So just looking at the slope of the linear part of that curve. And you can appreciate that for each one of these donors, there's again, a modest but significant increase in the rate of platelet accumulation. So these were two interesting observations, but we didn't really understand what was happening. And so fortunately, Aaron had created a really nice model of margination. So this is a direct numerical simulation of simulated red blood cells and green platelets that are moving over a porous thrombus. And so there is a kind of non-zero kind of Darcy type of flow through this porous medium. And so what Aaron and his group did is they simulated two different hematocrits. And so they look at a hematocrit of 0.4 and 0.6. And what you're seeing are little snapshots of these platelets. So every 2 milliseconds, they take a snapshot of the platelet and then calculate how long they spend within, essentially in very close proximity to this clot. And so what you can see, whether the thrombus is large or small, is that when you increase the hematocrit, you increase the amount of time that platelets are going to spend essentially tumbling and rolling over the thrombus. You can kind of correlate that to having a higher probability of sticking. And so that makes sense mechanistically that if they spend more time near the thrombus, they're more likely to stick to the thrombus. And that kind of helps explain the results we see in vivo and in vitro. So this is, I think one of my favorite examples of being able to work across all of these different types of models, both in vivo. We saw this reduced occlusion time in a mouse model of arterial thrombosis. In vitro, we're able to measure platelet accumulation at a very well-defined arterial shear rate. And then in silico, being able to really understand or be able to kind of back out that these residence times for platelets due to the kind of the compression of this cell-free region, which you can see in this cartoon, essentially allows more platelets to accumulate a little bit more quickly. So that's kind of one case study. Now I'll move to kind of going from the vessel scale to the cellular scale. And I'll talk a little bit about von Willebrand factor in more detail. But this is a large multimeric protein. It has a very large, you can think of it as a polymer that it's very polydisperse in the blood, and is unique in the sense that it's, I think, it's mechanically sensitive protein, at least in the blood. And so it circulates in this more or less globular format. It doesn't show up on here very well. And when it sticks to either collagen, so it sticks with high affinity to collagen 1 or collagen 3 through one of its domains. It also sticks to laminin, which is another very high abundant protein in the subendothelial matrix. It'll stick there, and it can be, because of the shear forces, it can become extended. And when it becomes extended, there's some cryptic domains inside of that globular format. So there's, in particular, the A1 domain. And the A1 domain is something that platelets want to stick to. And so there's a receptor on platelets, GP1B alpha, and that is a bond that will form and allow platelets to roll. Okay, so this is a very weak bond that forms very quickly. If you study leukocytes, it's somewhat similar to how leukocytes form bonds with selectin. So again, weak but fast, and so that allows platelets to roll. Once they're able to roll, they kind of can slow down. When they're able to slow down, and there's a little bit of also activation through that GP1B alpha, that activates a couple of integrins. So specifically on the subendothelial side, alpha-2 beta-1, which is an integrin for collagen, alpha-2b beta-3, which I mentioned before. It's this very high abundance integrin that can interact both with von Willebrand factor, but also with fibrinogen. And then there are some other integrins that have lower copy number, or kind of expressed. There's around 10,000 of each of these, but they all are important in different contexts because they can bind different proteins that are either in the blood or in other matrices. And finally, you get this activation. And so there's two very strong stimulators of platelet activation, glycoprotein 6, which sticks to both collagen as well as fibrin. It's a very powerful signaling molecule for platelets. And then CLEC2, which is a newly discovered one, but one that's pretty important in the development of the lymphatic system. It has a ligand called podoplanin. And these activate very, very significantly, and then you get all those other activation markers that I talked about, or activation states that you get before, granule release and exposure of phosphatidylserine. So here, the case study I want to talk about is something that we see clinically is von Willebrand disease. Von Willebrand disease is the most common genetic bleeding disorder. It has a pretty high incidence in certain ethnic groups, in particular in Northern Europeans. Sort of incidence in the United States is about 1 in 1,000. And there's different kinds of von Willebrand disease. I'll talk mostly about type 1. Type 1 von Willebrand disease is just people who have a mutation that causes a quantitative defect. That just means that they have a lower level of that protein in their plasma. That's the majority of cases. There are other types of mutations that fall under type 2, and there's many type 2 types of mutations, but these have defects that affect function. So they don't stick to collagen very well, they don't stick to the platelet receptor very well, several other mutations, and we'll get into them. And then there's a very rare type 3, which is these are people who express no VWF whatsoever. The symptoms of people who have von Willebrand disease is they have a propensity for bleeding, as you'd expect, but they bleed in an organ-specific manner. Mucocutaneous tissue is where people with von Willebrand disease are most commonly bleed from. So that's the nose, the gums, heavy menstrual bleeding, easy bruising are the presentation of von Willebrand disease. And there's a pretty, I would say, extensive differential diagnosis for von Willebrand disease. And so there's a number of tests. You measure antigen levels, which tells you, does somebody have a low level of von Willebrand factor? Do they have a defect in one of the functions of von Willebrand factor? And then you can go and look at these defects. How does it support platelet aggregation, collagen binding? Like I said, it's a multimer, and so you can look at the analysis of what's the molecular weights of the von Willebrand factor as well. And it can be treated either with desmopressin. Desmopressin is a drug that will help the endothelial cells release more von Willebrand factor. VWF is in the Weibel-Palade bodies of the endothelial cells, and you can kind of force that to be expressed with desmopressin. You can also give somebody sort of a replacement therapy, which would be giving them VWF typically derived from human plasma. Also, sometimes antifibrinolytics are used if somebody's going to have acute bleeding, like an oral surgery or something along those lines. And it's a very common disorder. And actually, the definition of von Willebrand disease has grown recently. It used to be that you would have to have von Willebrand factor levels of less than 30%, and now you can have von Willebrand levels less than 50%. So the number of people who we now say have von Willebrand disease is much larger. And even though there's been quite a bit of like genetic studies that can give you a significant idea of the heritability of this trait, the clinical bleeding is not always well associated with the VWF levels. And so we had done a study in our lab, and this is, I think, probably the most interesting disease, or at least the disease that I think lends itself really well to these types of techniques, because it depends on forces, both platelet adhesion and VWF function. And so what I'll show here are two videos. The ones on the left are just from a healthy control. The ones on the right are from somebody who has von Willebrand disease. And you'll see that first I'll show two images. This is at 150 reciprocal seconds. So this is a fairly low shear stress or shear rate that you would see in kind of the venous system. But you can appreciate that there's a really significant reduction in the accumulation of platelets, even at 150 reciprocal seconds. And if you go up to very high shear rates or arterial types of shear rates, you can see that really this person has no accumulation whatsoever with von Willebrand disease compared to the control. So this is a disease that really lends itself, again, to these biophysical approaches. This is just some images looking at somebody who has mild type 1. So this is somebody who has on the order of 10% von Willebrand factor, and then this is somebody who has no von Willebrand factor. And again, you can appreciate the sensitivity of this particular phenomenon to higher shear rates, where you really see, again, very little platelet accumulation if you have no von Willebrand factor at these higher shear rates. We can quantify this. This is all done in a kind of microfluidic device, and so we get these kinetic curves. We can look at how much of the surface is covered with platelets as a function of time, back out lag times, velocities or sort of rates of accumulation and how fast they go. But we can also measure response to therapy, and so DDAVP is sort of the acronym for desmopressin. And you can see this is this person who has like a moderate or low type VWF, and then we were treating them with DDAVP, and you can see the sort of rescue of their platelet function by just being able to secrete or have this drug that forces secretion of von Willebrand factor from endothelial cells. Okay. So now kind of moving from the cellular scale to the molecular scale, I want to talk a little bit about some of the models and different types of bonds that can be formed between a platelet and a receptor. Obviously, this goes beyond just platelets, but I'll talk about platelet receptors in particular. And the most common, or sort of the original bond model, this is the Bell model, if you're familiar with it, but it's what we would call a slip bond. A slip bond just means that as we apply more force, the lifetime of the bond goes down. So if I form the bond and I start to pull on it with a certain amount of force, the harder I pull, the faster it breaks, which is I think fairly intuitive. An ideal bond is one that essentially is insensitive to the amount of force, so the lifetime doesn't depend on the force that's applied to it. So slip bonds are often what we see with integrins. So like, for instance, it's been shown that the interaction between this integrin alpha-2b beta-3 and fibrinogen has slip bond-like characteristics. A catch bond has kind of the opposite effect. So a pure catch bond is one where the harder you pull on it, the longer the lifetime of the bond. So that's a little bit counterintuitive. And a catch slip bond is one that has elements of both, okay? And so that's something that forms essentially a peak. So it has a stronger lifetime until you get to some force. And then once you get to this peak, as you keep on pulling or you keep on applying more force, the slip bond behavior catches in. And so on platelets, the example of this is GP1B alpha, which I've talked about before, which is the integrin, or sorry, the glycoprotein. This is the receptor for VWF, and so you get this conformational change. It holds on, and actually, the harder you pull on it, the more it will, the higher the affinity it will bind with the von Willebrand factor, and then at some point you'll be able to unbind it. And so these catch bonds and catch slip bonds give slightly different behavior, and that's how, in the context of at least a platelet, different vessels, different types of injuries are going to have different types of forces. Different receptors are going to be at play or going to play different, let's say, relative roles depending on what the stresses are that the bonds are feeling. This is a classic experiment from Cheng Zhu's lab. These are platelet rolling experiments, and so a little bit like what I had shown before, but without collagen. These are just surfaces coated with von Willebrand factor, and then platelets are perfused over them. If you look at the right picture first, what you see is this, the behavior of the catch-slip bond in the rolling is kind of the inverse of the trend I showed before, is essentially that they roll slower, which you can interpret as the bond having a longer lifetime, until you get somewhere around, between 10, 20 dynes per centimeter squared. And then you get the slip bond behavior as you go above that. But this kind of shape is indicative of a catch slip bond. The fickle and non-fickle, that's just changing the viscosity of the fluid. And that's just done to show that this isn't a shear rate dependent behavior, it's a shear stress dependent behavior. So if you plotted this against the shear rate as opposed to the shear stress, these two curves would be offset a little bit. And so this just shows that it is, in fact, and the off rate of a receptor ligand interaction is always going to be dependent on the stress. The on rate can be dependent on the shear rate. The bond lifetime, and I'll kind of show here in the next few slides how you measure these, but this is just doing single molecule measurements where we measure the lifetime of the bond using a bioforce probe. I'll kind of allude to that a little bit later. Okay, so now a kind of an emerging concept in platelet physiology and in general, which has been around, I think, in vascular biology for a bit longer, but is becoming a bit more appreciated in platelet biology, is concept of mechanotransduction or mechanosensing, that there's actually a signaling that's occurring through platelet receptors that's mechanical, that doesn't depend on soluble agonists and depends significantly on force. And so in the context of what I'm talking here with platelet adhesion, we can see two different behaviors depending on shear rate. And so the first is what we see at lower shear rates, which I'll say less than 1,000 reciprocal seconds. And so we're looking at the interaction between GP1B alpha and von Willebrand factor, and then this integrin, alpha 2B beta 3, and fibrinogen. And if we have low shear rates, VWF maintains its globular conformation. And so what that means is that even though we can form a little bit of a bond here, we don't have access to a lot of these cryptic domains. So this A1 domain is buried inside of the globular form of VWF, and it needs to be pulled on to expose so that more of these A1 domains are available for binding. And so we don't really get a very strong bond here, but we do get tethering, or we do get rolling. So that's what we had seen before, is that if we get this rolling phenomenon, eventually we'll slow things down, and then we can get a little bit of an interaction between alpha 2B beta 3 pulling, and now we get what we call outside-in signaling, meaning the signal is coming from the bond on the extracellular side of the membrane. You can get some outside-in signaling, and so just by pulling on this integrin, you can actually, it's almost like this is a bit of a feed-forward loop where the integrin can bind, it gets pulled on a little bit, and that's going to create more of those integrins or more of those receptors in the high affinity state as a low affinity state. So that's mechanotransduction or mechanosensing through the integrin itself. If we go up to higher shear rates, so this is kind of goes up to the almost pathological range of say 10,000 reciprocal seconds, now we get this extended conformation of von Willebrand factor. It's rolling very fast, so you don't get engagement of the integrins. But there's a mechanosensory domain. So there's actually a domain on the GP1B alpha receptor that starts to come unfolded, okay? And when that starts to unfold, you get a signaling, this is a sort of circ-mediated signaling, and now what we can do is rather than getting this outside signaling that's like integrin to integrin, we get outside-in signaling that is through the GP1B alpha, through this circ kinase, and then interaction of the high affinity state or movement of the integrin to the high affinity state. So again, different receptors working in concert with each other depending on the mechanical environment. I mentioned a little bit about measuring bond forces at the single receptor level. This is a cartoon that shows something called a bioforce probe. So this guy here, this is it was kind of pioneered by Cheng Zhu's lab at Georgia Tech. It's a red blood cell on a micropipette. Then there's a polystyrene particle. Let me go back and play that again. Go. Yeah. Polystyrene particle that has the ligand that you're interested in. So in this case, it'd be the A1 domain of VWF. And then a platelet stuck on another pipette. And it can form essentially single bonds. And you can actually measure calcium measurements as well from here. So you're seeing, calcium is often used as a proxy for intracellular signaling. And through this, you can actually measure single bonds. And it's always kind of remarkable. And they essentially repeat this experiment over and over again, just putting them in contact, pulling them apart, putting them in contact, pulling them apart. And sometimes you'll get no bond. Sometimes you'll get a nice bond, and you can get this bond lifetime. And so that's how you make those you can get the distribution of bond lifetimes depending on how fast you're pulling. Sometimes you'll get some sort of bond ruptures. And from each one of these guys, you can kind of finally pull out and measure calcium as well. You can get a pretty good sense of what's happening at that single receptor level and get pretty good distributions where you can get a histogram of thousands or tens of thousands of events for a single integrin or receptor ligand interaction. Okay. So now I wanted to turn to another little case study and talk more about von Willebrand factor and its force-dependent functions. And so I talked about its this exposure of the A1 domain, which is what platelets like to interact with. It also exposes another one of another domain, a cryptic domain called the A2 domain. This is what regulates the size of VWF in the circulation. And so there's a enzyme called ADAMTS13 that circulates in the plasma. And what that does is it makes sure, physiologically, it makes sure that VWF doesn't get too large. And there's a genetic disorder called TTP. People who have TTP have a deficiency in ADAMTS13, and that means they have really large VWF multimers, which causes essentially platelet aggregates to form in the microvessels, and so you get this really significant kind of thrombotic occlusions, including multi-organ failure, just by forming these platelet aggregates. Fortunately, just very recently, there's now a recombinant form of ADAMTS13 that can be used to treat TTP. But in vivo, there's a number of things that can allow this to happen. Tensile force, which is what I've kind of been talking about when VWF binds to a surface and then gets pulled. When it gets secreted, like I mentioned before, VWF gets secreted in this from Weibel-Palade bodies, and when it does get secreted, it's quite large. We call it ultra-large von Willebrand factor, and so that needs to be chewed up a little bit. But also, turbulent flow by itself can create kind of this, and this is not, well, it's a bit controversial, I found, and maybe some folks here have a better feel for it. If there's turbulent flow in the human anatomy, I think it's either poorly characterized or maybe not very well understood. Certainly somewhere maybe near the heart valves and in some of the larger arteries. I don't think there's a lot of turbulent flow, but there can be a lot of turbulent flow in extracorporeal devices, things like vascular assist devices, ECMO, and other types of devices. And so we had sort of looked to study that, and people had shown that in the presence of platelets, if you put platelets and VWF in a rheometer and you shear them at a pretty high shear rate, you will start to get ADAMTS-dependent degradation. But we wanted to see whether you could get sort of degradation in the absence of platelets. And so we had to use a kind of a tool that I'd never used before, but we had found this worked pretty well. This is called a vein rheometer. It's a little bit different than the rheometers that I've been used to seeing, which are either cone and plate or annular rheometers that have like nice shear flows, but we were trying to get turbulent flows. And to get a turbulent flow, you needed a bit of a different geometry. And so what this does is, and this shows kind of as we increase the rotation rate, what happens to the torque, and you can see that there's like this almost like a asymptote above like I don't know, somewhere around 400 radians per second, 450 radians per second, where this thing goes from a laminar to sort of a transitional type of fluid flow to a turbulent flow at 450 radians per second. And so we did a little bit of CFD. This was in collaboration with David Bark, who's a professor at Washington University in St. Louis. And importantly, when he was doing these, he was able to find that the length scale of the turbulence was in places sort of micron scale, which might be relevant to the size of a really large VWF molecule. But what was kind of interesting is what we were able to show. These are on the top Western blots, and on the bottom what we call like it's a multimer, a different kind of gel. But I'll kind of walk through this, is that after 10 minutes, and this is laminar flow, transitional flow, and then turbulent flow. After about 10 minutes of turbulent flow, this is von Willebrand factor, you can see that it starts to get cleaved into these two bands that suggest that you're seeing some cleavage by atom TS13. So this is done in the presence of atom TS13. If we do it for 30 minutes, we see that, again, you can get some turbulent flow, not really any transitional flow or laminar flow. That doesn't seem to get it. If you don't put atom TS13 in, like the turbulence itself doesn't cause cleavage of the product. So just the turbulence with the atom TS13 tends to cause that. And then you can see this is the kind of distribution, and this is important. I probably could have showed this a little bit earlier, but this gives you a sense of what the distribution of von Willebrand factor looks like in a normal human. So there's a really significant distribution of different sizes. And when you go into these now in the transitional zone, even though we don't see cleavage on this Western, you can see there's a change in the distribution of the multimers as we move from laminar to transient to turbulent. This was done in the absence of blood cells, but if we do it in whole blood, we can see the same thing. Again, the turbulent flow by itself seems to be able to degrade von Willebrand factor, which is important if you're designing devices that are going to be in contact with blood. The other kind of part of this is that we get cleavage by the A2 domain, but we also get self-association by the A2 domain. And so VWF likes to form, this is how it forms multimers with itself, but it likes to form these strings with itself. And so that can happen at the vessel wall. It can happen between two platelets. And so here's a nice example from Jose Lopez's lab, where you can see these strings. So this is the VWF that's been secreted from these endothelial cells, and then you can see the platelets sticking on these strings of these guys. So it's they can form. They can be really quite long, like hundreds of microns if you let them form, and if you do it in the absence of atom TS13. All right, so for the last part of the talk, I'm going to get into, I think, my favorite subject here, which is something that I think is poorly appreciated and certainly comes out of a lot of work that Dr. Fogelson and I have done together, which is that blood flow itself, when people think about coagulation, they think about this biochemical network. Mostly in the, if we were going to model it and we were thinking about it, it's a big biochemical network. If I know the kinetics of what's happening, if I know all the catalytic rates and I know the binding rates, I should be able to predict what's happening, which you can to some degree. But when you put this when you have coagulation happening under blood flow, that creates another pressure or another kind of inhibitor, if you will, of coagulation, because you're constantly bringing in new, let's say, zymogens. So zymogens would be before these enzymes are formed. So I might say that, let me get my favorite one, factor 7 here. Factor 7A is an enzyme, but I'm bringing factor 7 in with the blood flow. So there's a supply of these factors by blood flow, but there's also a dilutional effect, because as I'm forming the enzymes, they're being carried away or can be carried away by flow. And so when I teach coagulation, because it's this gigantic mess of arrows, generally you can just think about it as there's three really important complexes that form. And so each one of these guys that are circled in red is a cofactor in the enzyme. So tissue factor is the cofactor for 7A, and that just makes the enzyme work a lot better. So when the enzymes are not bound to their cofactors, they're not very good enzymes. Their turnover rate is generally not useful. So the binding, with its cofactor typically makes that turnover rate 1,000 times, 10,000 times better. And so this is the one that we often focus on as the initiator of coagulation. So this would be the initiation of the extrinsic pathway in the classical coagulation cascade model. So this is often where coagulation starts. Tissue factor is expressed by endothelial cells, but much more strongly expressed by other cells inside of the vessel wall. So fibroblasts, smooth muscle cells tend to express a lot of tissue factor. Tissue factor can also be expressed by circulating blood cells. So monocytes, for example, can express tissue factor. So you can have tissue factor in the vessel wall, but you can also have tissue factor circulating. It can also be shed from many types of cancers. So, for example, pancreatic cancer tends to shed microparticles that have a very high tissue factor content. So that's one of the important complexes. The next one is what we'd call is 8A, 9A. This would be called the 10ase complex. And so it can form, it takes 10 and turns it into 10A. And so 8A is the cofactor and 9A is the enzyme. People who have hemophilia are missing one of those two molecules, either factor 8 or factor 9, or have a low level of them, I should say. And then prothrombinase is the other complex. So prothrombinase is the 5A, 10A complex, and that churns prothrombin and thrombin. Thrombin is ultimately what converts fibrinogen into fibrin, fibrin being this biopolymer that forms gives a clot its structural integrity. Thrombin can also activate platelets through a couple receptors. And so just like we use the Reynolds number to talk about what are the regimes of a flow field, when we are looking at these kinds of processes where we have mass transfer and we have reactions, there are two other useful dimensionless numbers that we can just calculate fairly easily based on, if we know the diffusivity and we know binding rates, then we can easily calculate this and at least know what zone we're in. So the first is, let's talk about the Peclet number. The Peclet number is just telling you what the rate of convection or the rate given, that's due to the flow divided by the diffusion rate. And so when this number is high, when Peclet number is high, it means convection dominates. That means if you want to think about, a Maxwellian demon sitting on a fluid packet, that molecule or that little Maxwellian demon is moving more or less with the flow field or with the streamlines. As the Peclet number gets smaller, diffusion starts to dominate, and we get something that looks a little bit more like a random walk. And so in every coagulation reaction, or anytime you have a reaction on a wall, both of these things are important because there's some finite boundary layer, and the boundary layer scales as the Peclet number to the 1/3. You have some zone, and in this case, everything that's below this black line is where we have significant amount of diffusion. And so this thing can get thinner and thinner, of course, as you increase the shear rate, but there's some area where diffusion is important. On the flip side, if we also want to look about the reaction regime, so we'd call this, there's many different kinds of Dom color numbers, so there's not one definition for it because it depends on what ratios you're looking at. But essentially, it's just something that says, what's the reaction rate versus the transport rate? And this is important because it tells us what's the rate limiting step in a reaction that's happening on a surface. So if we have a low Dom color number, the reaction is slow relative to mass transfer, so we would say it's reaction limited. If the Dom color number is high, then the reaction is fast relative to mass transfer, and we'd say something is mass transfer limited. And so it really kind of gets into, depending on which one of these Ks you're talking about, and this is why it gets very complicated very quickly. There's lots of different Dom color numbers. So you could write one for each one of these binding interactions. So there's an on rate, there might be some catalytic rate, there's an off rate, and then there's some mass transfer in and out of that zone. And often what we'll do is we'll just group that together as a single parameter and call it the mass transfer coefficient, or one kind of like what's the rate limiting step in those set of reactions, and we'll say that's the reaction rate. And then we look at this ratio to see whether something is reaction limited or transport limited. Okay, so this ended up getting a little bit blurry when we converted it over from my computer to here, but I think I can give you you can still get the takeaway message. So these are two experiments on two of those enzyme complexes. The one on the left is the formation of, so essentially converting factor 10 to factor 10A on the factor 8A, factor 9A complex. So on the wall is this complex, flow some 10 over it at different shear rates, and then measure how much 10A comes out. And so the bigger graph is the concentration as a function of shear rate, but the more important graph is actually the inset, which is the rate. So you can't read this, but it's like nanomole per second. So it's the rate at which 10A is coming off of the wall. And what happens here is that there's this transition where at low shear rates you're transport limited, meaning that this reaction goes faster, or you make more product the faster the blood flows or the faster the fluid flows. And so you see this increase in the rate of production as we get up to a certain shear rate. Once we get above a certain shear rate, we're no longer limited by how much substrate is there or how much factor 10 is there. And now we're completely reaction limited. So we can only go as fast as this reaction can turn over the substrate to the product. So that's true for this 8A, 9A complex. For the prothrombinase complex, or the 5A, 10A complex, it's always reaction limited, or at least always reaction limited, up to, a shear rate of, say, or down to a shear rate of, say, 10 or 50 reciprocal seconds. So in this case, it doesn't matter how fast the flow is, it's always limited by the reaction. You can look at this and you can kind of move up the scale. So if we look at fibrin polymerization, we see something somewhat similar. So now if I perfuse a little bit of thrombin into a flowing stream of fibrinogen and I measure how much fibrin has, I can use these two regimes, the mass transfer regime and the reaction regime, to get different kinds of morphologies in fibrin fibers. And so if I have something that's very reaction limited, I get this really thin film. So this is done on a membrane. So this is a polystyrene, or actually a Teflon PTFE membrane. And you can see these little films of fibrin forming. But eventually, if I start to move from this zone and I move up and I have higher and higher DOM color numbers, I can eventually form a 3D gel. And that seems to be what's important for forming a lot of fibrin. We can also do this through starting with plasma and moving all the way through the whole coagulation cascade. It turns out, and I tried to do this experiment a lot when I was a postdoc, that you cannot flow plasma over tissue factor and make fibrin under any conditions, no matter how slow you make the flow rate. You need something to protect the coagulation reactions from being diluted away and from the fibrin polymerization products to be diluted away. And so this is an experiment we did in our lab where we actually coated these, you can see these particles, we coated these particles with tissue factor. And if we put particles, and you can kind of think of them as like model cells, down on a surface, the interstitial space between these, if we have, you can maybe somewhat appreciate that these are pretty packed in here. Those were able to essentially allow all those reactions to go to somewhat completion, and you can actually form a bit of fibrin. And you can do that by, again, kind of going from a reaction limited to a transport limited regime, which depends, of course, on shear rate, but also in this case, a little bit on the geometry of where the clot's forming. Okay, so I talked about von Willebrand disease. The most common genetic coagulopathy is hemophilia. I had kind of talked about this or mentioned this before. This is folks who are people who are missing either factor 8 or factor 9. It's an X-linked recessive trait, so it affects 1 in 5,000 males all over the world. The ethnic distribution is pretty similar. It's important to note, though, that women who are carriers do experience or can experience clinically significant bleeding. So we often talk about hemophilia as a disorder that only affects men, but women who are carriers don't have the same presentation of bleeding that a male does who has a really significant deficiency, but they can experience clinically significant bleeding. But in men, and especially in boys, the way it presents is bleeding, and again, a very organ-specific type of bleeding. So the bleeding in hemophilia tends to be in joints. You can see this rather, swollen joint on this boy in the knees. So knees, ankles, shoulders, are areas, elbows, sort of the large joints are where you get these bleeds. And so you get a little bit of blood into the joint capsule. When red blood cells lyse, they release hemoglobin. Hemoglobin is a very inflammatory molecule, so you get inflammation. The inflammation causes more bleeding, and you have this pretty vicious cycle where if left untreated, you get degradation of all the soft tissue in the joints. You have to have a joint replacement surgery, or the joint just becomes completely immobile. The hemophilia is categorized essentially by how severe your deficiency is. So mild would be you have somewhere between 5 and 40% of normal levels, moderate 1 to 5%. People who have severe hemophilia have less than 1% or non-detectable levels of factor VIII or factor IX. And we have actually a pretty remarkable, even in my own career, like an incredible advancement in what we can treat or how we treat people with hemophilia. So since the 90s, replacement therapy has been the standard of care. So these are typically recombinant proteins of 8 or 9. They've gone through many generations so that their half-life is extended. In the United States and also in Western Europe, the standard of care now is prophylactic therapy. So we put boys on prophylaxis with these therapies when they're very young, even infants. But since then, there's been a number of other new drugs that have hit the market just in the last 20 years. They're often called bypass or rebalancing therapies. These are particularly important for people who develop inhibitors against the replacement therapy. So you can imagine somebody who doesn't have factor VIII or factor IX, you give them a recombinant form of that protein, they develop sort of inhibition, they develop inhibitory antibodies, then they have to go on to a different type of drug. So those drugs are very important for those people. And just in the last decade or so, gene therapy has started to make its way into hemophilia. And so we have the first trials in gene therapy were done in Europe. We're 7 to, I think, 9 years out from the first phase 1 trials. The first gene therapy product was approved in the United States just 2 years ago, so not a lot of people have had it, but a lot of people think this is kind of the future for treating hemophilia. But this is an example. This is also a work from my lab that maybe gives you a little bit of a sense of how blood flow and coagulation interact. And so these are images now of platelets and fibrin accumulating on a surface that has both collagen that supports platelet accumulation or platelet adhesion and activation, but also tissue factor that'll support fibrin formation. And you can see that when someone has a sort of mild deficiency, there's a significant decrease in the amount of fibrin that forms, but it will form around platelets. And remember, platelets are the sort of source or the catalytic site where coagulation reactions mostly happen. And based on our previous work, they also protect these reactions from being diluted away. The aggregates actually, we have the reactions happening in the interstitial space, we think, between platelets. But of course, if you have even a moderate severity or a mild severity, platelet accumulation is fine, and their platelets are perfectly able to adhere, but you can see there's none of the green fibrin here, so that doesn't form. And we've seen this again and again. This is actually older data, but we've been working in hemophilia now for over a decade. And it turns out that what we can, when we're looking at trying to discriminate people with different levels of factor VIII, the fibrin doesn't do a very good job at these lower levels. So you really can't tell the difference, in those last 2 movies. There's basically very little fibrin being formed, and so it's not very discriminatory on factor VIII levels. But we have found that platelet accumulation is actually a very good marker for factor VIII levels, and we see that here. We can actually distinguish severe and moderate hemophilia in this, and we can also look at responses to therapy. So we've done this with several different therapies, including some new bypassing agents. And it always seems that the platelets are a better signal in these assays than the fibrin is. We can also look at this is a model of hemostasis that we developed. And this also, I think this gives you a bit of appreciation for the mechanics of a hemophilia bleed as well. And so on the left is sort of a healthy control. On the right is somebody who has low factor VIII levels. And you can see that within a few minutes, you get this nice occlusive clot. This is, hemostasis. We don't have any blood going from the right channel into the left channel. But somebody who has hemophilia, they really can't form, they can neither form an occlusive clot, but this clot is, if you slow this down, and you can get a sense for this, the platelets just keep sloughing off. They really can't, you can imagine that as this the clot is getting larger, the shear stresses at this area are getting higher, and you're just not able to form a strong enough clot because there's just not enough thrombin being formed, and this thing essentially never occludes. It runs for, I think it runs for like 45 minutes, but it never occludes. So it gives you a good sense, but not only is this where you see a defect in coagulation really presenting as a weak or a leaky clot. And often when I talk to clinicians, they talk about weeping clots or clots that are very porous, where they don't ever really like form a strong clot. Okay. How am I doing? Oh, good. So I want to just end with just some emerging concepts and approaches. These are just my opinions, and some of you folks who are maybe starting in this field will take up some of these ideas. But one thing that is only really just beginning to be appreciated is the quantification and modeling the role of mechanosensing in platelets. And so I talked about the signaling through GP1B alpha and alpha 2B beta 3. It turns out platelets also express piezo1. Piezo1's of course a very well-known mechanical sensor on lots of cells, and this is just really starting to be. There's been, I would say, I don't know, half a dozen or so papers in our field that are starting to look at piezo1. So platelet mechanosensing, I think, is definitely something that's emerged as a new area to look at, and one that I think folks who are interested in modeling and biophysics really play an important role in helping quantify. Something I've been talking about for many years, and a lot of us have been working on, is do any of these assays have any clinical utility? Whether it's a microfluidic assay or a bond force measurement. And I would say that the results thus far are mixed. None of these things have translated into a clinical assay that I think adds a lot to how a clinician is going to treat somebody or diagnose somebody. Von Willebrand disease seemed to be the one that would be the most relevant because of the force-dependent function of VWF and platelets. But to be honest, there is a really robust set of assays for von Willebrand disease, and so sometimes I show this chart of all the assays that people do to kind of find the subtypes of VWD. I would say that's not a disease that's really in need of more diagnostic tools. I would say we have a lot of diagnostic tools for von Willebrand disease. So the translation of these assays, I think, has been maybe a little bit disappointing for some of us in the field, because it really, I haven't seen data that's been convincing, at least to me, that these are better than some of the existing assays that are out there. But I think more things are coming. I think we're sort of refining them a bit as well. A concept that we're sort of interested in is immunohemostasis. I know you're going to hear about thromboinflammation from Dr. Hunt later this week. And so that's the crosstalk between the innate immune system and the clotting system in the context of thrombosis. It was a very prominent part of COVID-19, or how some people with COVID-19 had clotting issues. Immunohemostasis is a little bit of more on the bleeding side of things. And so how does bleeding, how does the interaction of coagulation and platelets outside of the blood vessels happen? So this is a bit of an emerging concept. And then I think just in terms of approaches, I showed this one example of the elevated hematocrit. This is really what drives the field forward, is having teams of people or labs that can really move across the sort of in vitro, in vivo, and computational or in silico approaches, because any one of them I think can be very challenging. I think it's hard to build models that don't have good experimental inputs. It's difficult sometimes when you have experimental observations that you can't resolve what's happening. I think for us, working in coagulation, having mathematical models, working with Dr. Folgelson for a number of years has been absolutely essential for understanding what was happening, because we just can't track the sort of temporal evolution of hundreds of species that are changing at a millisecond or second time scale. So I think you guys are in the right places. You're at the right workshop, and I look forward to interacting with everybody over the course of the rest of the week. And with that, I'll take any questions.
Okay, thank you. Okay, thank you very much for this talk. Questions?
Jean-Christophe, hematology from the University of Montpellier and University Hospital of Nîmes. Well, first, thank you very much for this excellent presentation, of course. If I try to follow you, to what extent would we imagine treating hemophiliacs through inducing platelet hyperreactivity?
Treating hemophilia with platelet hyperactivity?
Yes.
Well, my intuition is that you don't want to have hyperactive platelets. Because even though, so maybe I'd like to take a step back and we think about people who with hemophilia are protected against some types of thrombosis, but not all. Not all. Sometimes, right?
I agree.
And so I think platelet hyperactivity, and frankly, even a molecule, right now, one of the targets for hemophilia treatment is antithrombin. And I think that's potentially a dangerous thing to be messing with. You may treat the hemophilia, the bleed in the joint, but if you end up causing thrombosis or a thrombotic kind of propensity on the other side, that might be a difficult trade-off for sure. I think that the approach that I've seen maybe along those lines that's kind of interesting or exciting is using platelets to deliver factor VIII or factor IX. So either through genetic manipulation of the megakaryocyte or actually, just ex vivo taking platelets out, loading them with factor VIII or factor IX, and then putting them in. That could be quite interesting, as a therapy. I think now we're approaching in hemophilia, there's almost so many therapies and so few people that I think that just the economics of hemophilia is changing very significantly. Where, I was just at a meeting last week. And we have anti-TFPI, we have anti-protein S, we have anti-thrombin, in addition to gene therapy. And this is a lot of things. And the drug there was a drug that was targeting protein C that was not pulled because of adverse events, but was pulled because the company decided this is too crowded. There's just not enough people with hemophilia to develop all these different therapies. So, yeah.
Okay, thank you.
Yeah.
Pierre Mongin, Strasbourg, France. Great talk, Keith. I have a couple of questions, and I really like you. You know about it, but the in vitro model of hemostasis. Mm-hmm. I heard what you said in the last slide, but are you not pushing it towards a device for diagnostics? Do you think it would be worth to do it or not?
Here's what I think if you think about the PFA 100. Mm-hmm. So there's a device called a PFA 100. I don't even know what that stands for. Platelet flow. I don't know what that stands for. Does it stand for something? I don't know. Anyways, it was a device, and it's a little disc with a hole in it, a little aperture, and it has on the inside of the aperture collagen or ADP or collagen and epinephrine, and it was a clinical assay, and people used this for this device for von Willebrand disease, for other platelet dysfunctions, aspirin resistance, and very few people use it now, right? And it was fun. Have you ever used one of these PFA 100s? No. We've used them, and we actually did a paper. Actually, the paper I showed with von Willebrand disease. We did our assay, the flow assay, and we did the PFA 100, and we did them together. And the PFA 100 was no worse than the microfluidic assay, and maybe a little bit better in some cases. It had a pretty tight distribution. So, sometimes I say, if the PFA 100 wasn't, and this is a pretty easy thing to use. You just put it's a very turnkey system. You get one number out, which is the time to occlusion. And I just wonder if that didn't work, why would this work? Because in a sense, it's wonderful. It has the microscopy gives you a lot of nice spatial and temporal resolution. I think it's wonderful for research. Whether or not you're going to be able to distinguish, meaningfully different, like clinical, predicting bleeding or be able to categorize people's bleeding or thrombotic episode. I don't know. These are hard things to do, I think.
Okay, great.
Yeah.
And then more in details about this system.
Yeah.
Sorry. Did you check the role of GP1B VWF compared to 2B3A fibrinogen in this device? Do you see differences?
We didn't look. We didn't look in terms of no, not in terms of blocking those receptors.
Okay.
Yeah.
Okay. Then can I last question? Okay. I really enjoyed the fact that you talk about the A2 domain of VWF, and you nicely showed on your slide like the two sides. So unfolding, and then it can be cleaved by Adams TS13, and unfolding it does self-association, and self-association probably fiber formation, and it becomes prothrombotic. So there are two sides, but with different outcomes. So how does it really work? I mean, do you think it's time dependent? Because I saw on one of your slides that you looked on the gels only after 10 minutes.
Yeah.
So self-association would be something very fast.
Yeah, I think it does boil down to kinetics and affinity, right? So if you look at the self-association kinetic, those affinities versus the affinity for Adam TS13 plus the kinetics for Adam TS13. Adam TS13 is a bit slower than the self-association of the A2 domain. So I think that it happens faster, but they're not. It's not the. I guess you have to get more granular to the amino acid level because that's not happening. It's on the A2 domain, but it's two different places, right? So even something can self-associate, be stretched, and still be cleaved, right? So yeah, I think it's a matter of balancing, looking at time scales and what are the relative affinities.
Okay, last question, if I may. So for VWF, there's caplacizumab targeting the A1 domain. Yeah. Works really well in TTP. But we know that in this pathology, what we want is increase the platelet levels, so avoid GP1B VWF interaction. But you can imagine if you use it in other pathologies, there might be bleeding consequences. What about targeting the A2 domain? Any idea? Did you know? Are you aware of people looking at that?
Targeting the A2 domain? I think Miguel Cruz has done some work along these lines of trying to target, or has peptides, maybe blocking peptides against the A2 domain. Yeah, I don't, but we haven't done any work in that field. Yeah.
Thanks.
Yeah.
Thank you. Other questions? The young people. No, you Maluk, you're not young anymore. Nobody? So I may ask to used to be young person.
Just a naive question for two maybe, but one to start with. When we speak about margination, is the margination the same in arteries and veins? First of all, that I don't know. And the experiment of the mice that you stuffed with red blood cells, in that case, you don't have aggregation because they don't aggregate, no? The mice blood. There is no aggregation per se, like the one described for humans.
Okay, so yeah, let me ask this second question first.
Okay.
So second question. So the platelets in the mouse model, they clotted faster.
They clot, but the red blood cells, for example, they are never in aggregates in rouleau and all what you described before.
This is a model in the carotid artery, so the shear stresses are very high. Whether you have more rouleau formation at higher hematocrit, perhaps like if you look in the venous valves or something, but it's hard. Mice don't. There's like one venous valve in a mouse. They don't have a lot of valves. They're very close to the ground. The first question about margination in arteries versus veins, my intuition is that the time scale at which it happens is going to be faster in arteries just because there's going to be more collisions.
So you say because of higher flow rates?
I think that the frequency of collisions will scale with the flow rate. But I think another, and people don't talk about it in veins. In veins, it's not something one thinks about or talks about very much. I will say though, so I didn't have this, another paper, this may or may not be relevant, but it was a model of venous thrombosis. And if we did it at low hematocrit, we never could get platelets into the venous valves. So this is just a model, a microfluidic model, not in vivo or whatnot. But if we had to have red cells, and we had to have red cells at physiologic hematocrits to get platelets into the valves, And then the platelets ended up helping propagate the clot through their expression of phosphatidylserine. So, yeah.
Thanks.
Hi. Thanks for the great talk, Simon Mendez from Montpellier. I had a question about closed loop effect, basically, because all the time when we're talking about, let's say, thrombosis or aggregation, we're local, we localize on one zone, but then, I mean, the platelets that leave that zone or the activated factors, in the circulation, they remain in the circulation. Yeah. So I was having a naive question about what happens then. Are there some deactivation mechanisms? And do we have some kind of closed loop effects that we have to worry about in some cases?
Yeah, I think my intuition about closed loop effects is that they're probably quite important. I mean, one example where they're clearly important is aortic stenosis. So in aortic stenosis, we know that we're getting VWF degradation, not because of turbulence, but we know that happens. And so a lot of times folks who have aortic stenosis have like bleeding because of their VWF multimers, but we also know that they have hyperactive platelets, and there's a number of other, diabetes, several other things where platelet hyperactivity is likely like a thrombotic risk. I think in the context, if a clot's forming and then a platelet's going through but not sticking, but maybe being exposed to some ADP or some thrombin and then coming back around, certainly it could still be activated. There are nitric oxide and prostacyclin, which are being secreted by endothelial cells, are supposed to, keep platelets in a more quiescent state, but I don't know of any, like in a human, like that kind of study, like a closed loop, right? I can tell you from our own work in vitro that if a platelet goes by a spot of collagen and tissue factor, and then you have another one downstream, it's much more likely to stick. So just that's a small, but just say millimeters away, certainly those platelets are more activated just by going by the previous clot. So whether that would, the time that your blood goes through your body is quite fast. So it's about a minute. So I think it's probably not unreasonable to think that there would be some effects along those lines.
There's going to be a vast dilutional effect. It's a small clot somewhere.
Yeah.
The blood turns to the heart, takes stuff there. What comes back? Sorry. I was suggesting that they may be important, but there's also a vast dilutional effect. If you have a small thrombus,
sure,
and stuff is shedding downstream, it's not going to come right back to that thrombus. It's going to go into the heart, get mixed with blood that hasn't been seen in thrombus, and the factors from the thrombus are going to be very, very much diluted. So that argues against a closed loop effect having a big role, at least in some contexts.
Right. I mean, but in a large artery, let's say this is carotid artery, everything has to go through it. Or not carotid, sorry, the aortic arch, right? Aorta. I mean, I understand your point. Yeah, I mean, even in the carotid, like what fraction of platelets that went through there are going to go back through there?
And there are also, I mean, certainly for the coag factors, there are things that will kill them off.
Sure.
Antithrombin and everything, and APC will. For the platelet side, not sure.
Yeah.
But there are clearance mechanisms too.
Sure. Yeah.
Jean-Christophe.
Well, another very general question and concern too. Would you have any idea of an ex vivo device or experimental condition practically allowing to study or to quantify the individual hemorrhagic risk?
Ex vivo hemorrhagic risk.
Yes.
I mean, in any kind of coagulopathy. Okay. Outside of our normal PT, APTT. Yeah. Okay. Yeah. I think there's an argument, a intellectual argument to be made that you want to treat whole blood. You want to do things with whole blood. because, you know, a hemorrhage could be anemia, you know, hematocrit is important, platelet count is important. You know, other dilutional effects of coagulation orders could be coagulation factors could be important. So studying whole blood, it's tempting, but it's dangerous. So this is what I've learned. Because I run, you know, I oversee this coagulation lab right now that does, you know, kind of molecular level diagnostics of different coagulation disorders. And this is what the way I answer this is I think you can screen, you can use whole blood assays, whether it's thromboelastography or as a screening tool to say risk, there's a risk here for hemorrhaging. I don't think you can make a diagnosis because how many different things could play out? You know, for example, like I work with some like trauma surgeons, and they really like using thromboelastography because it's point of care technique, and it's a whole blood assay, but its output depends on platelet counts, hematocrits, coagulation. You know, there's so many things, and then if you got that information, what are you going to do with it? Are you going to transfuse somebody? Well, I don't think you can tell from the output of that assay. I think that would be true for a lot of our assays as well, is that if you don't know blood cell counts, I think that's very hard. I think that's a very confounding factor. That's what I would say with whole blood. Even though you want to study the whole system. There's just, you know, the reductionist and sort of systems approaches are both useful.
Up.
Sorry, can't you? I don't understand why it is such a problem to work with whole blood, and can't you do both? I mean, count the cells in a tube and then elastography with the other, so that you can have both information and then analyze it.
Yeah, I think clinically, you know, these are become logistically. There's just a time associated with it, you know, so. In a trauma situation, you have no time, right? You are just making a decision in minutes. And I think that's at least my impression of why people like thromboelastography. You could do a CBC fast as well. It's not that it is impossible to do. I think that most emergency departments or trauma places, they don't have all these assays to make this assessment. So I think certainly you could do a CBC. You could do a TEG or some other whole blood type of assay and have some algorithm that says, you know, let's just say I have a system and there's some contribution of hematocrit, there's some contribution of platelet count, there's some contribution of all these factors. You know, how complicated does that model have to be like predictive? And then if you have, you know, I'm always worried about overfitting. If I have some linear regression that has 16 parameters in it, you know, it could be hard.
Thank you very much for this overview and this lecture. And I have a more general question, more like a curiosity from like a beginner to an expert, about the microfluidic used like a tool to investigate platelet functionality and whatever. Do you think that can be like a useful tool also for diagnostic, but also like to ask, do you think like there is at the moment like a sort of lack of standardization in the microfluidic field, for example, so sure. Aspect ratio of a channel can influence, or like just the length? For example, when something is very length, it's complicated to have a fully controlled from the entrance to the exit of the flow. And the second question that maybe could be like only one answer is if the microfluidic tool can be used like also in the field of circulatory support, like to recreate what happened in LVAD or ECMO in something like in a microfluidic system.
Yeah, so I think for the first question, is a very insightful and excellent question. And you know, Pierre and I have worked on this for a long time, and it's a major, I don't think it's a problem. Some people think it's a good thing, but everybody does this differently. You know, everyone has, when people are making their own devices in their labs, everyone has a different size channel. Everyone puts down proteins differently. Entrance effects can be important. So when you read the literature, it can be, I think it's very hard to compare studies because no two labs do it the same. Now there are commercial tools. There's one called TTAS that's from Japan. There's one called Bioflux, it's in the United States. There's like a lot of people I know use these Abiti chips they're from here. I think what's missing in the field is somebody to do, and these are hard things for like labs to do. Someone has to do a large study, and they need hundreds of people with some well-defined condition, whether it's von Willebrand disease, and they need to compare the output of that device to these other assays to really answer this question. I don't think it's impossible that it's useful clinically, but someone has to do someone has to do those studies, and they have to be kind of big. And I think this is why it's hard for an academic lab to do them, because I can't do 200 people with von Willebrand disease. I mean, it would take me 10 years to get that many people. So you need, you need almost like, what's similar to when you're doing a clinical trial for a drug, or you're trying to do a large scale devices. You need a lot of centers involved, and you need a lot of folks to say, this is the protocol. We're all going to draw blood into sodium citrate, and we're all going to use it. All the pre-analytical things matter. When do you use it? How long did you wait? Did you do it at room temperature? Did you do it at 37 degrees C? All those little details matter, and I think in a research setting, people aren't so concerned. I mean, I run my assays my way, and Pierre does his way, and we don't talk about it. But if you did want to do, I think that's what's kind of missing in the field is just, you can look, every paper will have 10 patients in it, right? But in mine included, and if we did an assay, we've done something with like 20 people, 30 people with hemophilia, but I haven't seen anything where someone's done hundreds of patients, and I think that's what you need, and you need to do, you have to compare it against the gold standard that exists right now. So whether that's, ristocetin-induced platelet aggregometry for von Willebrand disease, something like that. So you can say, okay, there's value added here. And then what's difficult in bleeding, and I'm not as familiar with thrombosis, but in bleeding, quantifying clinical bleeding is extremely hard. So that's the other issue, is that what is bleeding and what's important bleeding. And so in hemophilia, we count joint bleeds, how many bleeds somebody has in their joint per year. And that's fine, but that's not all, that's self-reported. These are all just surveys based on people who have hemophilia and they have a joint bleed, or how much factor did they consume, could be another one. But that's super specific to hemophilia, which is a rare disorder. Something with von Willebrand disease, which is more common. We have like a survey. There's a survey called the Bleeding Assessment Questionnaire. And you just fill out, it's like 100 questions. How many nosebleeds did you have? Heavy menstrual bleeding? Did you bleed during childbirth? All these kinds of things, and then you set it up. But it's not, it's a questionnaire, and ultimately there's some limitations to that. So I think that's another part that's difficult, at least on the bleeding side of things, is that it's difficult to quantify clinical bleeding. And so even when we've done these assays, ultimately, just comparing two assays to each other isn't very satisfying. You want it to be compared to the relevant clinical output, and for bleeding that can be a little bit hard. And did I answer both your questions? Okay.
Oh.
Yeah, I wanted to go back to the question before from Professor Lee, who asked whether we have devices that give information on the bleeding risk. I would say we don't. So I agree with Keith. I guess we have lots of assays, whether they are preclinical, using every day in the lab, that gives a lot of information on hemostasis. Sorry. We also have diagnostic tools, TTAS, PFA 100, or thromboelastography, and the thrombogram assay as well, that gives information on coagulation, on platelet function, etc. But we don't have any tool that gives any information on the bleeding risk. And so the question whether these devices will be able to do it, I doubt it. Because even just one missing piece is the vascular system, right? Sure. And then it's not there, so it plays a really important role, because at the end of the day, what is really the bleeding risk, right? It's at one stage a vessel that is opening probably and that is bleeding. So if we now go to small animals, I mean, if you have, let's say you develop a molecule and you want one day to do a phase 1 in humans, you first have to go to animals. And so the one that we are using a lot in the lab are mice. But there we have exactly the same problem. We only have models, like a tail bleeding time assay, that gives information about hemostasis and not the bleeding risk. Sometimes you can add lots of and high doses of some of molecules that play a major role in coagulation and or regulate down the platelet function, and you don't have any bleeding, so they're not spontaneously bleeding. Right. So over the last couple of years, some of the lab developed model of inflammatory bleeding in the brain, in the skin, in the lungs. So we're getting a little closer, but just a little closer.
Yeah, the point about the vascular wall is obviously one that should have we should have mentioned is the interaction between the endothelium and coagulation and platelets is obviously central. And our, of course, microfluidic models that people do this, but we do this in my lab. This is not something that's a clinical assay. I mean, you're growing endothelial cells inside of a tiny channel. It takes 4 days for them to be ready. So they're wonderful research tools. Yeah, it's hard to see some of these things being clinically translated.
Any other questions or comments? I have one. Regarding this connection between the size of the VWF and the turbulent flows, the turbulent regime, do you have any explanation for that? Because it looks strange because the energy that you have at those very small scales is essentially zero. I mean, you don't have anything at Komogorov. So could be the mixing, that you better mix the LMT13 and the VWF due to turbulence, or could it be what?
I guess, if you think of maybe in a little more of a sophisticated manner about what is a globular protein, right? It's not just a static thing. It's very conformationally dynamic. And one thought is that the frequency, the sort of the turbulence, something that's happening at pretty high frequencies, is maybe just allowing a little bit of, if you're pulling on that string of yarn, it might just be allow it to kind of pull it out a little bit more than maybe it does, under normal circumstances. I don't think it's a mixing effect. It could be.
Yeah, if you want to put it on, to put it, I mean, you need time, but the Komogorov scales are very short time, and so I don't. Well, it looks strange.
It is an interesting observation. We've seen it not only in this rheometer, but also in there's actually a kind of cool assay that you can do just in a well plate. If you shake a well plate, and we've done this with a high-speed camera and actually watched the flow, so you can create a little turbulence just in a well plate. You can get the same effect. But you can't do it no matter how if you do it in a cone and plate rheometer or a rheometer where you have a laminar flow. No matter how high we went up, if we went to very high shear stresses, we never saw any cleavage.
Okay.
Yeah.
Okay.
Yeah.
All right. So I believe it's time to close this session. So thank you again for your very excellent introduction.